Okay, so I'm just going to review the process of DNA replication and protein synthesis so that you have a good understanding of why the processes occur and where they occur and how they occur. Okay, so the first thing that you need to understand is that we are all composed of cells, okay? And the cells have lots of organelles in them that do certain things, and we know that there's Golgi apparatus and mitochondria. We have some ribosomes. We have endoplasmic reticulum. We have vacuoles. We have lots of different organelles inside of the cell, and each of those organelles does a certain job. We also have a nucleus, and I'm drawing it quite big because I want you to understand the purpose of it, okay? So let's just take a look. We've got endoplasmic reticulum, and that's for packaging things for the cell. Typically, if it has ribosomes on the side, it's for packaging proteins. That's rough ER. And if it's smooth ER, it's for packaging fats. The ribosome is to make proteins. The Golgi apparatus is another organelle that's charged with packaging things, and actually it sends things into out of the cell, it packages them so that they can fuse with the membrane and send things out of the cell. The mitochondria is the powerhouse. That's where the ATP is made. And we've also got vacuoles for storage, just like your garage. The cell stores things, okay? So I think I better make my drawing a little bit smaller. So we need to know all of those things. Inside of the nucleus, we have our DNA. And typically the DNA is all in like a big string, sort of like your Christmas lights when you go to get them at Christmas time. The year before, you were so busy that you just put them all in a box, okay? So this is what the cell will typically look like on during the regular basis. And don't forget, we've got the plasma membrane, or the cell membrane, and I'm talking here about an animal cell. And the plasma membrane allows things to come in and out of the cell. And don't forget, it's made of a phospholipid bilayer. And in here we have the cytoplasm, which is the jelly that makes sure everything stays in its place. Okay? So there's your cell. Now, we learned that at some point the cell will want to divide and make two daughter cells. And cells are constantly dividing because we need to make new cells all the time. Cells don't live forever. They die quite often. They get to the point where they're no longer able to perform their function. So what they do is they get ready to make two new cells that can perform the function. So as it gets older, it produces two new cells. And that is the process of mitosis, where you end up with two daughter cells with exactly the same things in it as the parent cell, okay? And it makes all of these things so that each daughter cell has the same ER and vacuoles and Golgi and mitochondria, okay? So how does it do that? Well, let's take a look at the cell, and let's take a look at the DNA, okay? So remember, I told you that normally the DNA is all wound up in a big mess, sort of like Christmas lights. When a cell is getting ready to divide, what it does is it organizes that DNA into nice tight coils called chromosomes, okay? So chromosomes are made of DNA, it's just tightly coiled DNA. So instead of looking like this mess, it looks nice and tidy. Sort of like when your mom asks you to clean up your room. you take all those clothes that are strewn all over the floor and you fold them up into nice tidy piles, okay? So those are chromosomes, which are just tightly coiled pieces of DNA. Now, if I took those chromosomes, let's use a different color here. Let's say I take one of these chromosomes, okay, and I'm just going to enlarge it here. Okay, so there's my chromosome. And chunks of each of those chromosomes are called genes. And each of those genes is a chunk of chromosome, which is a large piece of DNA. Now, if I take that and unwind it so that I just have my DNA, we know that DNA is double-stranded and it's got phospholipids, phosphate and sugar backbones, and it has nucleotide bases. And each of those nucleotide bases actually is a code for something. So let's say this gene codes for, a specific protein, let's say lactase. So in your DNA, you have a gene that has a message to tell your body to make lactase. And if I take that gene and stretch it out, I am going to see that it has one strand and a complementary strand. And this strand has certain bases on it. I don't know the code for lactase, but I'm just going to make it up. A, T, C, G, G, A, T, T, A, C, G, C. Okay? And you have to know that the complementary strand to that piece of DNA is T, A, G, C, C, T, A, T, G, C, G. Okay? So each piece of DNA is double-stranded. Those genes have specific letters that tell what the gene is supposed to be. Okay? So this is the message or the instructions to make this protein. And don't forget, lactase is an enzyme, and enzymes are proteins. Okay? So now I have this piece of DNA. Okay? So that should tell you the difference between a chromosome, DNA, and a gene. So let's take a look. So a chromosome is a tightly coiled piece of DNA. A gene is a section of DNA. And DNA is a nucleic acid that is in a double ladder or double helix inside of the nucleus. Okay, let's continue on. Let's say a cell decides that it wants to make two daughter cells. Okay, this is mitosis. So I need the cell to replicate its DNA. So it has to take its chromosomes and double them, because you want to have twice as many chromosomes as the parent cell. So the parent has to make 2 copies of its chromosomes, so that when it splits into 2, each daughter cell gets 1 copy of the parent's chromosome. Okay? So it has to make copies of itself. Okay? So in order to make copies of itself, it has to replicate its DNA. Okay? Now, DNA replication is actually quite simple. So let's say I have DNA. I'm just going to draw a short strand, but this, you guys got to remember, there's lots of DNA in your cells. Okay? So let's say I have A, T, G, C, A, which means I have T, G, C, A, T. When this strand wants to make another copy, what it does is it has to split in two. And we have a protein or an enzyme called DNA helicase. And helicase comes in and unzips that double strand. So then you end up with a replication fork, which is where it opens up. And then what you have to have is DNA polymerase has to come in. And DNA polymerase comes in and makes a second strand on each of those. Okay? So then it'll make the complement here. And the complement here. Sorry, I'm squishing it in here. So now you have two brand new strands of DNA from the parent strand. Okay? Now, so each of these is called semi-conservative because you conserved one part of the original strand, and then you have one new strand. Semi-conservative. One strand of the original and one new strand. So now you have two copies of the parent DNA. And now each of these copies can go into the new cells. That's DNA replication. Now you have to remember that these base pairs are added by DNA polymerase. Which goes in and adds the nucleotide bases. You have to know that. You also have to know that it goes in a 5' to 3' direction. And because these are called anti-parallel, this end is 5' and this end is 3'. So one of those strands is called the lagging strand because the DNA polymerase has to go down to this end and start adding the bases. And it can do it right away on the leading strand. You also have to know that these little pieces or chunks are called Okazaki fragments, and they are glued together by DNA ligase. Okay, don't forget ligand is a base word for putting together, gluing. Don't forget that's the glue. Okay, this is the guy that adds the bases. These are the little pieces. And DNA polymerase cannot operate without our friend RNA primer. Okay, that's the guy with the checkered flag. He actually goes along here and says, start here. Okay, so he puts a little checkered flag, which is a piece of RNA. Put the checkered flag in so that it knows where to start. Okay, okay, so that's replication, DNA replication. That's making a copy, making a replica, replication. Making a replica, a copy of the original strand. Here we go. We make two copies because we have two daughter cells. Okay, the next thing that you have to know is making proteins. The whole point of DNA in your cells is to make proteins. And proteins are the things that give you characteristics or help your body perform functions. So once again, we go to the cell, and inside of the cell there's our DNA. Okay? I'm gonna leave everything else out of the cytoplasm except